Hello students, welcome back. Today we'll see conjugation, that is in your 503 paper, unit number 3. Different methods for transfer of gene is included, out of which we'll start with conjugation. So first of all, what is conjugation? It is a natural gene transfer process, or you can say it is a sexual transfer method of plasmid where two cells are involved. Between two cells, a bridge is formed, and via that bridge, DNA is transferred. So, it is a natural gene transfer process where horizontal transfer of DNA occurs. Why it is called sexual transfer? Because two cells, one is a male cell and another is female cell. Between this two cell, transfer occurs. That is why it is called sexually transfer method. This mechanism of transfer is plasmid encoded. That is, this transfer is maintained, or you can say it is regulated by plasmid coded genes. Here, physical contact, that is cell to cell contact, is required between a donor male cell and a recipient female cell. Donor male cell have F plasmid, that is fertility plasmid is present in donor male cell, while F plasmid is not present in recipient female cell. That is why it is called F negative cell. So physical contact between this donor and recipient cell is formed via a bridge, which is also called a conjugative tube. Donor is called sexually positive organism as F plasmid is present, while recipient, a female cell, is called sexually negative organism as plasmid, that is fertility plasmid is not present. This ability to perform transfer, it is confirmed by a set of genes called fertility genes, which are present on F plasmid. That is, this fertility genes exist on a circular double-stranded DNA which replicates independently, or they can be integrated into chromosome. Integrated form is called episome. So plasmid, we know what is plasmid. Plasmids are circular extrachromosomal DNA which can exist independently. It can replicate independently, or plasmid can integrate into bacterial chromosome, and that is called episome. Bacteria containing these genes are called male cell. That is, the bacteria which have F plasmid that have fertility factors, they are called male cell. Such male cells form pilus to attach to the neighbor female cell. The two cells then are drawn together and the DNA is transferred from one bacteria to another. Now once this pilus or conjugative tube is formed by male cell, it attaches to the neighboring female cell, and that cells are drawn towards each other, and horizontal transfer of F plasmid from donor male cell to female cell occurs. This method was discovered by Lederberg and Tatum in 1946 in E. coli. So, in short, what is conjugation? Conjugation is a transfer of genetic material between bacterial cells of opposite mating type, that is a female negative cell and a male positive cell, that are in a physical contact with each other. Or you can even say it is a process in which DNA is transferred from a bacteria donor to a recipient bacteria by cell to cell contact. See, you can see over here in diagram, this is a positive male cell, this is a negative female cell. Here you can see this is a bacterial chromosome which is of course present in both the cell, but F plasmid, that is fertility plasmid, is only present in male cell, but it is not present in female cell. So your name is given male bacteria A, female bacteria B. Pilus is formed. This bridge is formed by male cell. Transfer of genetic material from bacteria A to bacteria B, that is cell to cell contact via pilus or a tube occurs, and this plasmid is transferred from donor to recipient, and at the end of this process female cell, that is F negative cell, also becomes positive That is, it becomes male and it even can transfer its F plasmid to other female cells. Who is donor? The cell which contains F plasmid, they act as donor, and that is why it is called F plus cell. While who act as recipient? F negative cells, they are called female, they act as recipient. So F plasmid have fertility factor, or you can say a sex factor, or you even can say transfer factor, which allows the transfer process. So first, as I told you, cell-to-cell contact occurs. Here you can see in diagram a sex pilus or a conjugative tube, or you can say bridge pilus is formed between, it is formed by plus cell, which it's attached to its neighboring female cell. Here you can see in diagram. So formation of F bridge occurs between two cells. Then transfer, that is once attachment or pilus is attached to the female cell, then genetic transfer of single-stranded DNA occurs. That is double-stranded F plasmid, which is present in male cell. Single-strand F plasmid will be horizontally transferred to female cell, and after that both the single-stranded plasmid which is present in male and female get replicated, and double-stranded F plasmid is present after that in both the cells. So first first step was cell to cell contact, second step is transfer of single-stranded DNA, third DNA synthesis. That is, a complementary strand of DNA are replicated in both F minus and F plus cell. So single-stranded plasmid, now replication occurs, and in both the cells double-stranded plasmid will be present. Target DNA is incorporated into F genome. So at the end of this process, the target F plasmid, which was first not present in the female cell, now it is incorporated into female cell. Then separation. Cells are separated. It forms two F plus cells with the completed recombinant plasmid. Both serve as donor cells for further conjugation. So after that, the pilus formed is the pilus which is formed break down. The cells are separated, and at the end of this process, both the cells become male F positive cells, and they are ready. They can donate F plasmid to another female cell. So this is the complete process which is shown in diagram. The first step, that is pilus formation between the pilus formation is formed by male cell and attachment occurs. So this is male cell, this is female cell. Pilus is formed. Next, both the cells, they come in a close contact with each other. Then single-stranded cut occurs in F plasmid by enzyme, and one strand of F plasmid is horizontally transferred to another cell, and at the end both the cells are male cells. Next term is HFR. What is HFR cell? When F factor, that is plasmid, exists in an integrated state with the host chromosome. See here it is given in diagram. F plasmid, here it is independently present, that is independent from chromosomal DNA. But when this F plasmid, it gets integrated into host chromosome. So these are called HFR cell, high frequency recombinant cell. When F factor or F plasmid gets incorporated into host chromosome by recombination, such cell are called HFR cell. So HFR conjugation. So this is the process of HFR conjugation. Here, this is F plus cell. When F plasmid is incorporated into host chromosome, it becomes HFR cell. So this HFR cell, when it performs conjugation, this is a female cell which is not having plasmid. So here F genes are present. So pilus is formed, bridges formed between two cells. And what occurs? Here you can see single-stranded cut occurs and single-stranded DNA is transferred from positive to a negative cell. Then portion of F plasmid partially moves into recipient cell. Conjugation ends with pieces of F plasmid and a donor DNA in a recipient cell. Cell synthesize complementary DNA strand, and at the end, donor DNA and recipient DNA recombine, making a recombinant F negative cell. So here, complete transfer do not occur. Some of the portion of F plasmid and some of the portion of host chromosome is transferred to negative cell, and that get replicated and is converted into double-stranded DNA, and that portion which is incomplete F fragment as well as some of the portion of host chromosome of donor is transferred to recipient cell, which can be integrated into host recipient chromosome, and finally a recombinant is formed, which is again F negative. It is not F positive because complete F plasmid is not transferred from donor. So here you can observe in F plus F minus conjugation, at the end F plus was converted into F minus female was converted into male at the end, but here in HFR and F minus conjugation process, female, that is F minus, is not converted into male at the end. So F plus means free plasmid, independent plasmid is present. It can be converted into HFR by integration process, which is a reversible process. It can even be excised out. So in this free plasmid, that is F plus, it turns F minus into F plus by conjugation process, while HFR will not convert a negative female cell into plus at the end process. So F plus and HFR formation is reversible. F plasmid can be integrated and it can be even excised out. When the F factor reverts from integrated, a free state, it may sometimes carry with it some chromosomal DNA from the adjacent site of its attachment. That is, such an F factor is known as F prime, or you can say F dash factor. Now, in this process, once HFR is formed, and from HFR, again that F plasmid is excised out. During this excision process, some of the genes of chromosome is also taken away. That type of plasmid, or you can say that type of F factor, is called F prime. That is, along with F plasmid, some portion of host chromosomal DNA is also present. So when conjugation of F prime with female F negative occurs, that process is called sexduction. When F prime conjugates with a recipient female F negative, it transfers along with the F factor the host DNA incorporated with it. So as F prime contains host chromosomal DNA also, so during sexduction process, along with its own gene, host genes are also transferred into female F negative cell. So this was the process of conjugation. Thank you.